Transformation of bacteria is a very useful tool in modern science, used to introduce exogenous genes into the cells. There are many transformation methods for bacteria, two of the most common being electroporation and chemical transformation. These two methods were performed in order to compare the efficiency of chemically competent cells and electrocompetent cells. Transformation is a process in which a cell is altered so that it can acquire and express genetic material obtained from the surrounding medium or environment. The most common use for transformation is the introduction of recombinant plasmid DNA into bacteria. The bacterial strains can either be naturally transformed or can be made competent by artificial means. Transfection, on the other hand, is the introduction of foreign DNA into the nucleus of eukaryotic cells by non-viral methods. The most common gene transfer methods are chemical, lipid, and physical methods. There are many different methods to transform cells. However, not every method can be applied to both prokaryotic and eukaryotic cells. To transform eukaryotic cells, electroporation and sonoporation methods are commonly used. The electroporation is a physical transfection method that consists in applying of electric pulses to create temporal pores in the cell membrane. It can be used in bacteria and eukaryotic cells, and it's the most efficient technique. Sonoporation can also be used as a transfection mechanism and is an ultrasound mediated by cell membrane permeabilization or introduction of membrane pores using ultrasound. The pores of ultrasound are delivered by a machine and they can be of a selected intensity and duration depending on the sample. This method has been used in bacteria, fungus, yeast, plants, and mammalian cells. There are several things that must be considered in order to select a bacterial transformation method. First of all, it is important to consider the methods through which the cells have become competent. Another thing that should be considered is the cell membrane and wall composition, because some cells' competence can be resist to certain transformation methods. We have to consider too, before choosing a transformation method, it's the size of the plasmid that will be introduced into the bacteria. Electroporation is a good method of high molecular weight vector transformation. And finally, it is also important to make into consider the capacity of cells to resist the damage caused by the transformation method in order to ensure that the cells will be alive after the transformation process.
For chemiocompetent cells, once you have obtained the pellet, you get rid of the supernatant and then you resuspend it in 10 millimeters of CaCO2.1 molar cold, and then you keep it incubating in ice for 10 minutes. Then you centrifuge, then you get rid of the supernatant and again resuspend the pellet in 2 millimeters of a solution of CaCO2.1 molar and 15% of glycerol. Then you incubate it for 10 minutes in ice. After that, you make 200 microliters aliquots and you store it at minus 70 degrees. For electrocompetent cells, once you get the pellet, you get rid of the supernatant. Then you resuspend the pellet in 10 millimeters of a solution of glycerol at 10% cold. Then you incubate it in ice for 10 minutes and then you centrifuge again. Then you get rid of the supernatant and resuspend the pellet in 2 millimeters of 10% glycerol cold. Then you make aliquots of 200 microliters and you store it at minus 70 degrees. For chemiocompetence, you take one of the aliquots of the chemiocompetent freeze cells. Then you place it in ice until it's get unfrozen. Then you add 5 nanograms of an unknown plasmid. Then you incubate it in ice for 10 minutes. Then you give it a thermic shock in the thermoblock to 42 degrees for 45 seconds. Then you give it another shock, put it in ice for 5 minutes. Then you add 300 microliters of soak medium in sterility conditions and then you incubate it for 37 degrees during 30 minutes with agitation at 250 revolutions per minute. Then you spatulate 2 volumes of the different dilutions of the culture in LB plaques with ampicillin. You let it incubate it for 16 to 20 hours at 37 degrees. After that, you must count the colonies that were grown and you will calculate the yield of the transformation. For electrocompetent, you need to first place the electroporation cell of 0.2 centimeters sterile in ice. Then you take an aliquot of the electrocompetent cells and you place it in ice until it gets unfrozen. Then you place in a 1.5 millimeters tube a volume of 40 microliters with a mix of the bacteria and 50 nanograms of a plasmid. Then you transfer 40 microliters of the such said mix to the electroporation cell and you place it in ice until you program the electroporator.
In the petri dish of the electroporated cells, we could observe no growth, while in the petri dish of the chemiocompetent cells, we could observe the growth of 234 CFUs after 17 hours. The transformation by electroporation did not work, and this could have been because the voltage was not adequate. It could have either been too low and it did not transform the plasmid, or too high and it killed all the bacteria. The chemiocompetent cells gave a higher yield, but it was still relatively low. All of the CFUs that were observed were white. The low efficiency of the CFUs of the chemiocompetent cells could have been because they were observed after only 17 hours after they were plated, instead of 24 hours as stated in the protocol. To conclude, it is necessary that in a genetic engineering laboratory, we can generate competent cells that can replicate and express the genes that are inserted in them. It is necessary for cloning vectors and generating libraries, as well as obtaining recombinant proteins. It is necessary to check the electroporator since it isn't normal to obtain such a low yield.